Okay, so thank you, Chris, for the introductions. Good morning, everyone. So I'm Karen Laserna, and today I will be presenting on the introduction to the metabolomics approach and its applications. So this is the first part of the three-part webinar series that we have on metabolomics. And for this webinar series, we'll try to answer your questions regarding metabolomics. So we'll try to cover what is metabolomics, why should you consider using it in your research, when and where can it be applied, or in what areas of research can it be used for? And lastly, we'll cover the how, or how can we perform a metabolomics experiment. So for this session, we'll cover more of the what, the why, the when, and the where. In the subsequent sessions, I will be talking more about the hows of metabolomics. So we'll cover the experimental and the practical considerations when performing a metabolomics experiment on March 13, and we'll cover more of the data analysis aspects of metabolomics on March 19, which is basically on how do you use chemometrics in metabolomics data analysis. So first, we cover what is metabolomics. So yung metabolomics, actually, mahati mo yung term into two different words. So you have metabolome or metabolite, metabolism, and omics. So first, we talk about metabolism and metabolites. So metabolites refers to the totality of the cellular activity. And as you can see from this diagram, it is a highly uh, coordinated network. And this is also actually very highly regulated. So it consists of different reactions of chemicals, which we'll talk more about later. And these network consists of smaller systems or multi-enzyme systems that are known as the metabolic pathways. So these metabolic pathways can perform different functions. So it can be for purposes of generating energy. It can be for conversion of nutrients to endogenous molecules or molecules that you find in the body. It can be to build larger molecules from the monomers or the building blocks. And it can be to break down toxic substances as well to more to metabolites or compounds that can actually be excreted from the body. So just now I have already mentioned metabolites. So metabolites can, in this case, makes up your metabolic pathways. So they are the substrates and the products, the intermediates of the metabolic pathways. And these metabolites are actually small molecules that are produced in or by biological processes. So they are in the molecular weight ranges of 50 to 2000 Dalton, and they can be endogenous. So when we say endogenous, these are metabolites that are produced within the biological system. So this can be primary metabolites. So primary metabolites referring to those that are part of the primary metabolism of the biological system, such as your amino acids, your carbohydrates, your lipids, organic acids, etc. In the case of some other organisms like your plants, you will have your secondary metabolites, which are actually metabolites that are produced in response to either stress or as a part of their defense mechanism. You also have exogenous metabolites. So exogenous metabolites naman are those, so if you have synobiotics, which are like foreign agents that are introduced into the body, or drugs, they are also metabolized by the biological system. And these are what you know, or what is called as exogenous metabolites. So here are just some examples of these metabolites. So you have phenylalanine, which is an amino acid. You have glucose 6-phosphate, which is a phosphorylated version of glucose, a well-known molecule for most of us. And strychnine is, of course, an alkaloid, a secondary metabolite. And here also is acetaminophen glucuronide, which is acetaminophen, your paracetamol, that is metabolized into the glucuronide form. So, the next part of the metabolomics term is your omics. So, kapag sinabi natin bang omics, ano ba yung omics? So, when omics is used, uh, it is used to reflect totality. It is used to reflect the use of high throughput technologies in order to simultaneously and comprehensively measure certain molecules. So, depende na lang dun sa kung ano ba yung nasa first part of the term. So for example, when we say genomics, it is to simultaneously and comprehensively measure genes of a particular biological system. When we say transcriptomics, it will be to measure transcripts, gene expression. When we talk about proteomics, it will be to measure proteins. Glycomics will be to measure carbohydrates. Metabolomics, of as what we're going to cover now, is to measure metabolites, and lipidomics is to measure lipids. So as you can see here, from the cascade, from the molecular cascade here, you have your from the chromosomes, you have your genomic expression, and down closer towards the phenotypic expressions, you have more of the smaller molecules such as your lipids, metabolome, or the metabolites, and in in this part, glycomics will be more covering on the post-translational modifications of your proteins wherein they can be glycosylated. So yun naman yung sa part ng glycomics. So metabolomics, when the term was first coined in 1998 by Oliver et al. When they measured the changes in metabolite concentrations with deletion or overexpression of a gene. So they were looking at deletion, the changes in the metabolite concentration with the deletion and overexpression of genes in yeast experimental model. The term metabolomics, on the other hand, was first coined by Oliver Finn in 2002, when he talks about metabolomics as being the comprehensive analysis wherein all metabolites in a given biological system are identified and quantified. Now, another group also came up with the term metabonomics. So this is actually coined by Nicholson et al. In 1999, and this is defined as the comprehensive and simultaneous systematic profiling of multiple metabolite levels and their systematic and temporal changes caused by factors such as diet, lifestyle, environment, genetic effects, and pharmaceutical effects, both beneficial and adverse in whole organisms. So medyo mas comprehensive yung definition ng metabonomics. And in some cases, during the early parts of this metabolomics era, medyo i-interchange siya, and in some cases, some would say that, okay, metabonomics is subdivision of metabolomics. On the other hand, yung iba naman sinasabi that metabolomics is subdivision of metabonomics. So kumbaga, yun, there is a lot of contribution in the terms, but eventually ngayon it's more of metabolomics that is being used. And just to sum everything up, when we say now metabolomics, it basically refers to the simultaneous and comprehensive analysis of metabolites in a given biological system under study. So in order to do metabolomics, you need to make use of certain instrumentations, and these are some of the main instrumentations that are being used, particularly more of the MS-based and the NMR instrumentation. So you have the spectroscopic techniques or spectroscopic instrumentation mainly used for purposes of fingerprinting, whereas MS-based in the NMR spectroscopy, which actually can allow you to identify metabolites, are more for the different purposes. So MS-based metabolomics can make use of either direct injection or coupled with separation techniques such as liquid chromatography, gas chromatography, as well as capillary electrophoresis. So we'll talk more about these instrumental aspects when we go into the experimental considerations and workflows of metabolomics in our next session next week. So why should we take the metabolomics approach? So we have your genomics and proteomics, which are the more established and older levels of omics approaches. So now, why should we consider using metabolomics? So first, we have to consider yung similarity of metabolites across different species. So just think about it. Um, for genes and proteins, there can be differences. There's a lot of diversity between genes and proteins across the different organisms. Whereas for metabolites, glucose, for example, in pigs is glucose also in humans or in other organisms like in microbes, in plants, glucose is glucose. Whereas in genes and proteins, there can be the same, for example, the same gene or protein may provide a different function in different organisms. So there can be diversity in that. Whereas for metabolites, it is highly conserved. You can also consider yung proximity niya to the phenotypic expression. So not all genes and proteins get translated to a phenotype. So when we talk about phenotype, this is yung actual manifestation of the biological system, kung ano yung nakita mo, kung ano yung state of the biological system that it can be observed. And yun nga, so in genes and proteins, not all gene and protein expressions are translated into the phenotype, basically because you have post-transcriptional modifications and post-translational modifications that occurs, and these are not captured. Kasi yung mga yun is nangyayari after ng gene expression and after the protein expression. So these post-transcriptional and post-translational modifications, as well as environmental influences, are actually captured by the metabolites or in the metabolomic expression. So metabolome is closer to the phenotype. You can also expect that from the gene expression, there is also a magnification of the changes as it goes to the metabolite level, and this provides increased sensitivity in terms of like detections. Metabolome can also reflect effects of environmental factors, health condition, diet, gut, microflora, host interactions. So lahat yun nakaka-capture in the metabolome, and this can provide you with a better, in a sense, more accurate representation of your phenotype in terms of the molecular aspects. And lastly, it has easier translation to diagnostic and monitoring devices and assays. So I'm sure a lot of you or all of us are familiar with the use of glucose. So glucose as indicator or a biomarker of the development of diabetes, for example, and you also have your creatinine measurements that serves as a physiological monitor for the development of renal dysfunction. So alam natin na kapag medyo may problema yung kidney, tumataas yung creatinine, and kung merong diabetes, mataas yung sugar. So these are just a few examples of the use of metabolites to indicate disease or physiological condition. So meron ng utility. Even before the time of metabolomics, there is already the utility of metabolites for these purposes. So this is just to illustrate kung the numbers involved when we talk about the different omics levels. So in genomics, you have about 40,000 genes. Transcriptome, you are dealing with around 150,000 different transcripts. Proteome, you have over or around 1 million proteins that are currently detected. And for in terms of primary metabolism, you have about 7,800 metabolites. But this is, of course, not including secondary metabolites in plants and microbes, which is in the case of plants can reach up to about 200,000. So mostly in terms of the functions, mas known yung functions of the metabolites compared to like the level of your proteomes, transcript, and the different genes in terms of like effects of environmental influences, mas reflected din yung environmental influences in your metabolites. Now, this is of course not to say that metabolomics is the end all and be all among the omics levels. So this is definitely not the case. Actually, in the same way as you look at a map, so di ba yung mga maps natin, actually, if you look at Google Maps, you can actually divide a map into different layers. And you can consider or see your different omics levels as different layers of that map. And only in making use of the different layers are you able to um, see a better picture or a better view of the overall picture. So if you make use of these multiple layers, so mas maganda or mas comprehensive yung cover mo na picture. And so usually, in order to better understand biological systems, there can be a need to make use of one, two, or more of these omics levels. And I think for the applications later, as well as in the applications that will be shown in the subsequent sessions, you will see this integration of different omics in different studies. Okay. So when can we use the metabolomics approach? So metabolomics can be used to characterize a particular phenotype. So this phenotype can be a disease state, for example, or if you are looking at wild versus mutant plant species or knockout models of certain diseases. So you can use metabolomics to characterize them. It can be used to assess the effects of a treatment. So it can be, if you're trying to assess the effects of drugs, of certain pollutants, or in the case of like stress. So if you are looking at the stress response of, for example, plants, so you can use metabolomics to assess the effects of those treatments. It is also used to identify potential biomarkers. So when we talk about biomarkers, so this, an example again of this will be your glucose and creatinine, which are respectively used for the monitoring of the development of diabetes and renal dysfunction. So yun, you can make use of metabolomics to characterize different disease, and from there on, try to identify metabolites that can be indicative or can be used for early diagnosis of these disease states. And lastly, it can be used for metabolite fingerprinting. So when we talk about our fingerprint, di ba? So yung fingerprint, it is actually unique per person. So in the same way, like different organisms, different biological systems will have a unique fingerprint that can be obtained using its chemical composition. So in this case, We can look at the metabolite fingerprint of the individual, of the organism, and these can actually be used for purposes of authentication, establishing the origins of a particular organism or product, and it can also be used for purposes of quality control. Okay, so where can we use the metabolomics? So ito naman, we'll talk more about the actual research fields and areas and the examples of the types of studies that metabolomics is actually used in these areas. So this is by no means exhaustive. I'm sure you still have a lot of other uh, research studies and research areas that you may be working on that are not included in the next slide. If you see the potential of the use of metabolomics in your research, please do let me know if in any way I can be of help. And we can explore how to use metabolomics in your particular research area. Okay, so when some of these applications of metabolomics will be, of course, in plant biology, when looking at transgenic research, what are in terms of like changes of the metabolic profile of these transgenic modifications. It can also be used to characterize different breeds. It can also be used to study the mechanisms involved in plant pathogen interactions. It can be used to assess the stress response of plants, and it can be used as part of the functional genomic studies. In the sorry, in the case naman ng food and nutrition, it can be used to establish host microbiome interactions. Particularly if you look at the gut microbiome, so your gut microflora, yung mga microorganisms or microbes that you have in your gut also produces their own set of metabolites. And actually, this can also be reflected in our own metabolic profiles. So, and in addition to that, pwede mo tingnan yung effects of changes in the gut microbiome and how this affects the metabolic profile of the host. It can also be used to personalize nutrition. So, dito po papasok yung mga current um, so dito papasok yung mga current trends in terms of like personalization of like, for example, medicine, personalization of nutrition, of diet in reference to like your particular genomic characteristics. So dito papasok yung mga nutrigenomics, the use of nutrigenomics, and together with that, you can make use of metabolomics to characterize your the persons or the different persons. So you can also use it for nutritional assessment. So nutritional assessment, because of course, your endogenous metabolites are actually good measures of the nutritional needs of a particular person, especially in the case of studying malnutrition and what are the needs for supplementation. It can also be used for purposes of authentication and quality assessment and control. So dito papasok yung making use of metabolomics, metabolite fingerprinting for purposes of authentication and quality control. In terms of natural products research, currently, as we have covered in our recent Tuklas Lunes program, fellowship program, you can make use of metabolomics in the replication in drug discovery. So instead of going through the usual classical approach, one way or as complementary to the more established ways of doing drug discovery research, you can make use of metabolomics to identify, okay, in terms of like, you can correlate your assays. So ano ba yung mga uh, different components or fractions that are bioactive, and you can make use of metabolomics to determine ano ba yung mga metabolites that are present in those fractions. It can also be used to identify the effects of these natural products in the person or in the organisms that consume them. So yung, for example, your herbal products, herbal traditional herbal medicines, what are their effects in the body? And this can be characterized using metabolomics. And again, making use of metabolomics in terms of metabolite fingerprinting, it can be used for purposes of quality control. Kasi as we know, natural products are very comprehensive in terms of like components, complex mixtures. So herbal products can make use of metabolomics approach to characterize or establish yung the profile of a good quality herbal product, and from there it can be used for purposes of quality control. And of course, it can also be used for authentication. So I think in some cases, particularly in traditional Chinese medicines, there can be adulterations, or especially if the contents are quite expensive, there can be cases of adulteration or total substitution um, of other products, and you can make use of metabolomics to characterize the metabolic profile of an authentic product. In the field of agriculture, you can make use of metabolomics for assessing crop improvement, for quality assessment of crops, for the assessment of growth conditions and treatment effects, as well as to establish geographic origins. So, for example, crops that are of the same variant that are grown in one area will have a different metabolic profile compared to crops that are grown in another area. So you can make use of these metabolic profiles to distinguish ano ba yung produced in one area and what is produced in the other area. Sorry. Okay. So in terms naman of environmental research, metabolomics can be used for toxicological assessments and to in exposure studies to establish kung ano ba yung effects of these exposures and toxic compounds in the biological system. Lastly, we look at preclinical and clinical applications of metabolomics. So it can be used for, again, purposes of biomarkers, for physiologic monitoring. It can be for diagnostic biomarkers. So it can be used to diagnose the presence of certain diseases. It can also be used, for example, there are some metabolites that can be used to assess the effects of certain treatments. So maybe one of these could be your glucose. So if your diabetes treatment is working, then of course you can expect that your glucose levels can go down. In the same case for creatinine, if your kidney, renal drugs or kidney treatments are working, then you would also expect your creatinine levels to also go down. It is also very commonly used in order to study disease mechanisms. So metabolomics has been used in large cohort studies such as your Framingham heart study, as well as your in Europe, they have this UBioPred asthma study where they make use of metabolomics to characterize the molecular phenotype of asthma as well as cardiovascular disease. You also have your, it can also be used to identify drug targets in pharmacology to assess the effect of drugs and to establish whether safe ba to take these drugs. So in some cases kasi, actual manifestations of like problems or complications with the use of certain drugs can be coming much later. and hindi kagad siya makakapture in the physiological state or in the phenotype of the person. So it is good to make use of metabolomics to monitor these different metabolites kasi this can be indicative of like perturbations or bad effects of certain drugs. So kapag nakita natin that there are certain metabolites that are more associated with development of renal dysfunction or development of liver dysfunction, if you see that with the treatment of or with the use of certain drugs, tumataas na yung mga metabolites na yun, then it is a time to consider whether to continue using yung drug or to see whether the dose can be lowered and so forth. So these are again different applications of metabolomics. So these are basically more of those that come to mind for in my case. But as I mentioned earlier, this is in no way exhaustive. And I'm sure you guys have a lot of different studies and use and can make use of metabolomics in your research. So please do let me know if there's any way that I can be of help and assist you in your research on how to make use of metabolomics. And so in the next part of this presentation, I will give you snippets of how I used or how our group has used metabolomics in different projects. So I'll give snippets of 2 projects that I have worked in collaboration with students as well as with collaborators in NUS. And also I will give a more detailed presentation on an application that I have been particularly involved in terms of a preclinical model. So for the first application that I'm going to show, this is the work of a collaborator, Song Shuang and Srishti, together with their professors, Wang Chiwa and Yu Tan. And the health services, HSA organization of Singapore, so Health Services Authority. So, sorry. So for this application, they made use of biochar for urban agriculture, and they wanted to assess the effects of how this biochar application can have on the bok choy. So pak choy or brassica rapa is, I think, also known as Chinese cabbage. I'm not very sure, but this pak choy is a very common vegetable that is present and highly consumed in Singapore. So a lot of these urban farming studies has been in making use of this vegetable for the studies. So biochar is actually produced in the pyrolysis of horticulture, municipal, and agricultural wastes. And this has the characteristics of being lightweight and more durable. So actually in urban farming, so yung characteristics naman ng soil in urban areas are not very suitable for farming, di ba? So and usually you make use of compost to make the soil more suitable for farming. But compost will degrade through time. And so an alternative to that is to make use of biochar. So yung biochar is, again, more lightweight, more durable. It can last for a longer period of time in maintaining your organic content of the soil for purposes of farming. So we made use of NMR and LCMS for the untargeted metabolomic profiling or untargeted metabolomic analysis of the bok choy samples. So these were extracted using organic solvents and subsequently analyzed. So these are sample spectra in terms of like in NMR. So this is proton NMR spectra as well as in LCMS. So LCMS, we made use of a high resolution QTOF LCMS system that is capable of tandem MS also. But this is mainly showing you the TOF MS scan that is produced. We made use of both positive and negative ionization. So again, details of these experimental considerations and parameters we will cover in the next session, which will be next week. So after we have obtained the data, we subjected the data into chemometric analysis. So chemometrics is it involves statistical techniques that allows for the simultaneous analysis of different variables. So usually when you work with untargeted metabolomics like these, you generate a lot of data. And in order to make sense of those data, you make use of chemometrics. So we'll again cover more of that in the third session. But for now, this is just to show you that, okay, we have used chemometrics in the data analysis, and one of those chemometric techniques is the use of principal component analysis. So principal component analysis allows you to see patterns or trends that are present in your data. And in this case, you will be able to see clustering of the different replicates for different treatments. So merong walang added na biochar. So 0% biochar. You also have samples with 30% biochar, and you have samples that has 50% biochar. So you can see from here, merong clustering of those that are of the 30% biochar, and those that are grown in the 50% biochar, and meron din for the 0%. So in order to identify ano ba yung mga variables or metabolites that has caused this differentiation into these different groups, you have to look at what is known as the loadings plot. So in the loadings plot, you will be able to see which variables influence the differentiation or the separation of these different groups. So usually, in expressing or in interpreting the score plots and the loadings plot of principal component analysis, you have to consider same locations will refer to which features are higher in that group of samples. So in this case, for the 30% biochar, those that are on the right or on my right in the plot will be those that are high in your 30% biochar. So those that are on the upper left quadrant will be those that are high in the 0% biochar, and those that are in the lower left quadrant will be those that are high in your 50% biochar. So the variables here are actually the chemical shift values, or the ppm, the chemical shift in ppm values of your NMR spectra. So this region actually covers more of the carbohydrates. So from the spectra, yung mga values na to actually covers the carbohydrate region of the proton NMR spectra. And this is just to show that with application of 30% biochar, there is an increase in these metabolites, mainly in carbohydrates. On the other hand naman, those that are on the lower part are actually corresponding to some amino acids. And again, this shows that certain amino acids, um, those that are on this region are those that are corresponding to the amino acids. And actually, that means the level of amino acids are higher in your 0% biochar, and the application of biochar results in a decrease in the levels of these amino acids. Now, this is more of the LCMS results. So you look at, first we look at the results of the LCMS analysis in positive ionization mode. So here again, you see some clustering or some differentiation of your samples for 0%, 30%, and 50% biochar. And mainly what you can see is that in terms of the loadings plot, you have an increase in certain flavonoid glucosides for your application of biochar. So for 30% and 50% biochar, you have increased levels of these glucosides, flavonoid glucosides. On the LCMS negative mode naman, again, you see a differentiation of your 0% biochar from your 30 and 50% biochar. Merong mga outliers na kasama dito, but mainly the other replicates are present more on the right quadrant of the score plot. And these are actually influenced by the increase in levels of your some of these flavonoid glucosides, as well as your glucosinolates. So glucosinolates like gluconapine, glucobrasylcanapine, uh, progoitrine, etc. So actually those values here, the number values here correspond to your MZ value, which is your mass to charge value, that can be used for the identification of your compounds based on accurate mass. Again, we are using a high resolution QTOF LCMS system. So you'll be able to use the accurate mass generated for purposes of identification. And we also make use of tandem MSMS. So we make use of information dependent acquisition, MSMS, for purposes of fragmenting your these precursor ions, these molecules of interest. And then from there, you'll be able to confirm ano ba yung identity talaga ng mga ions na to. So, lastly, this is just to show the effects of biochar treatment in the camphorol biosynthesis. So from the different loadings plot here, you can see that mainly it is the camphorol glucosides that are camphorol and its glucosides that are mainly perturbed in terms of like the pathways that are affected for biochar application. So you can see here which metabolites are higher in the 0%, and subsequently which ones are higher in your 30 and 50%. So this is just to show that with the application of biochar, there is increased glycosylation and acylation of your camphorol metabolite. So actually, in this particular project, we were able to see the complementarity of making use of LCMS and NMR for metabolomic study. You see more of the primary metabolism changes in the NMR side, whereas you see more of the secondary metabolite changes in the LCMS analysis part. So again, this is one application. Another application that I would like to briefly share with you is the use of metabolomics to study the metabolism of tri and butyl phosphate in the earthworm Perionyx excavatus. So tri and butyl phosphate, or TBP, is actually it's a ubiquitous compound. It is commonly used as a component of plasticizer, industrial solvents, and it's also used as an anti-foaming agent. So you can expect it to be present in different environmental matrices, such as in soil, in water, in the air, etc. And so we made use of this earthworm model. Perionyx excavatus is a common earthworm that is found in Asia, in tropical regions like Singapore. And we used untargeted LCMS metabolomics, again, making use of the high resolution QTOF LCMS for purposes of getting high mass accuracy MZ values, as well as tandem MS information for purposes of later on identifying the different metabolites of these, or this xenobiotic, the TBP. So these just shows like using untargeted LCMS-MS, we were able to identify certain metabolites of TBP. So this is just showing you the use of MSMS to identify, to confirm the identity of these TBP metabolites. So you can see from the structural elucidation, making use of the fragmentation pattern that is obtained. And we were able to identify some of those metabolites, such as the phosphorylated TBP, hydroxylated TBP, ethanol TBP, and the sulfated ethylene glycol DBP, or dibutyl phosphate. And actually making use of the relative of the quantitative information that you can get from the LCMS, we were able to obtain relative quantitation of the levels of these different metabolites in the earthworm. So we have looked at acute exposures of 1/10 and 1/5 of the LC50 dose for 2 days, and also at the chronic exposure of making use of 10 milligram per kilogram and 50 milligram per kilogram exposure that is incorporated in the soil. So yung acute exposure, we made use of filter paper contact test. On the other hand, yung chronic exposure naman, the TBP was mixed into the soil, and the earthworms lived in that soil environment, contaminated soil environment, for 28 days. So as you can see from here, mainly the uh, metabolites that are higher in levels in both um, acute and chronic exposures are mainly M2, M3, and M7 and M8. So this corresponds to these metabolic path or metabolic routes, which are mainly the hydroxylation and the phosphate conjugation of your tributyl phosphate. So this just shows, actually, we wanted to emphasize with this research that when you're studying these xenobiotics, you would need to consider the different metabolic products, or the exogenous metabolites of these xenobiotics in the terms of like the risk assessment and safety assessment. Kasi you wouldn't know, there is a possibility that some of these metabolites, or the transformation products of these xenobiotics, can actually be more harmful or more toxic than the main xenobiotic compound. So this is again more of an application of metabolomics in environmental research. Now we go to a more details. We make use of I will show you more details in one application of metabolomics in my thesis. Actually, I studied mainly the metabolomics of trauma injury for my thesis, and this has been done in collaboration with the Defense Science Organization, National Laboratories of Singapore. So they are the Defense Science research arm of Singapore, and it has been a privilege working with them. So for this particular presentation, I will present on the metabolomic study of complex combat trauma injury in a porcine model. So porcine model will be a pig model. So trauma injury is one of the leading causes of morbidity and mortality worldwide, particularly for people under the age of 45. And as you can imagine, this is the age wherein people are at their prime. They have significant contributions to the society, and this results in significant personal, social, and economic impact kapag nagkaroon ng trauma injury. Now, after a trauma injury, there you can envision like this particular illustration here. If you throw a pebble into the pond, you would expect ripples to radiate from the point of impact outwards. And the same goes for trauma injury. So there is a complex network of changes that occurs after a trauma injury. And this can affect organs that are far from the site of injury. So for example, the injury can be just in the head, um, because of certain physiological or pathophysiological situations or effects. Um, the complications can go up to other parts of the body, such as your kidney, your liver, your heart, etc. So again, these can be normal physiological changes that can proceed uncontrolled, which because they proceed uncontrolled can cause complications, or they can be pathological changes. So pathological changes mainly, for example, the presence of infection, sepsis, and so forth. So one of these normal physiological changes that can be harmful if it proceeds uncontrolled is the inflammatory response. So when we talk about inflammation, di ba, right away, pangit yung notion natin. We think of inflammation as a disease in itself, or as a bad symptom in itself. But actually, inflammation is a response of the body to a particular insult or injury. And it is meant for wound healing and regeneration. So when an injury occurs, there is usually a local response at the site of the injury that is facilitated by cytokines, endothelial cells, and macrophages. And this activates a series of reactions and highly controlled processes in order to restore homeostasis. Now, if the homeostasis is restored, there is resolution and recovery. But if the reactions or the processes proceeds uncontrolled, this can result into systemic inflammatory response syndrome, or SIRS, and subsequently it can affect different organs, and these can eventually lead on to multiple organ dysfunction syndrome, or MODS. So as you can see, the inflammatory response is actually, you can consider it as a double-edged sword. So on one hand, it can be useful. It is actually an integral part of our wound healing response. But on the other hand, if it proceeds uncontrolled, it can lead to a whole slew of different complications. And so from here, you can see that if an initial injury is not resolved, this can proceed to SIRS and affect remote organs and cause multiple organ failure or dysfunction. So in combat trauma, there is a higher risk of mortality, of course, and the nature of wounding agents are more deadly kasi nga this is combat trauma settings, and so the weapons are definitely for purposes of like disabling or killing the opponents. You expect multiple wounding in a combat trauma settings of it can be from explosions or from gunshots. It can be trauma from car collisions, etc. And there is the presence of persistent threat. And lastly, you would expect that in a combat trauma setting, you have limited resources. So hindi available in lahat ng resources that are usually available in a hospital setting. And so there is a need to really study combat trauma injury in order to identify ano ba yung mga pwedeng gawin in terms of like treatment strategies, as well as supplementations um, for combat trauma injuries. And This is also in order to address problems of delayed access to definitive care. Ano ba yung pwedeng ano ba yung pwede mong i-implement na initial treatments at the combat trauma scenario before they reach the hospital in order to prevent these different secondary complications. So the mechanism of combat trauma injury is usually coming from fragments of explosives. It can also be second to that will be because of like bullets. And in terms of the primary site of injuries, these are usually to soft tissue. So more of your, for example, the abdominal area, your internal organs, as well as your extremities. So these are the primary sites of injury. And you can see a prevalence of penetrating trauma in combat injuries. Now, with penetrating trauma, so once, for example, a gunshot or impact of explosives fragment, this can result to serious injury, trauma injury, that can lead to blood loss. So deficiency of oxygen reaching the organs can lead to shock, and this can trigger severe metabolic derangements and a complex inflammatory response. Eventually, this can also lead to the development of SIRS and possibly MODS. And so, for this particular study, we made use of a porcine model, pig model, to study complex combat trauma based on a complex combat trauma model that was established by Cho et al. And why did we use pigs? So actually, pigs are more, in terms of metabolism, physiology, and hemodynamics, pigs are actually uh, similar to humans. So that is why we made use of pigs for this particular model. And this is just to show you a diagram of what has been done. So for the trauma group of pigs, so you have male Yorkshire, I think Yorkshire landrace pigs that are um, uh, subjected to trauma, and you have a group of pigs that are not that are subjected to the different experimental procedures, but not the injury phase of this trauma model. So for this model, initially, so of course you have your anesthesia um, um, treatment, your intubation, instrumentation, and subsequently, before the injury phase for the trauma sample, but blood sampling was obtained for both the sham and the before trauma to serve as their baselines. And then the injury phase consists of penetrating trauma using a bolt gun. And then subsequently, blood is drawn out in order to induce or to simulate blood loss. In this case, they looked at 60% blood loss for this model, and then subsequently subjected the animal to hypothermia. So they reduced the temperature, the core temperature of the animals to around 33 degrees centigrade, and subjected them to a 30-minute shock period, which is usually the time period that is considered in terms of like um, the time by which the injured can get some definitive some care or uh, um, uh, treatments in order to relieve or to address their injury. Okay, so this is again a combat injury simulation phase. So with this combination of injury, you expect trauma hemorrhagic shock to develop, and afterwards, blood is again obtained in order for us to later on be able to to compare the metabolic profiles before and after trauma in comparison to your sham, which has been subjected to all the different procedures such as anesthesia, intubation, instrumentation, but except for this injury phase. And in this manner, we'll be able to identify ano ba yung metabolic changes that can be associated with trauma injury. So this is to show the instrumental analysis workflow. So we have plasma samples obtained from the pigs. They were subjected to extraction and protein precipitation using solvent for the supernatant. This was divided into aliquots for helic or hydrophilic interaction, liquid chromatography, MS analysis, as well as reverse phase LCMS analysis. And a certain portion of the plasma sample was used also for NMR analysis. So this is high resolution proton NMR analysis as well. And subsequently, the data was extracted and analyzed using chemometrics. So again, we will cover more aspects of this instrumental workflow or the experimental workflow for metabolomics in the subsequent session. So this is just to show you an overview of the data processing that has been done. So we have done some data preprocessing and extraction, and subsequently this is subjected to chemometric analysis. You have multivariate analysis such as PCA and OPLSDA. And then because there are so many metabolites that are involved, we further narrow it down using univariate statistical analysis, using Wilcoxon-Mann-Whitney for fold change in order to see fold changes in the metabolic profiles. And we made use of certain databases for the identification of these metabolites. And subsequently, we did metabolic pathway analysis and also correlation network analysis. So this is just to show you an example of the NMR spectra of the plasma metabolic profile of trauma injury. So this is to compare your before sham and after sham, as well as before and after trauma. So in terms of like the visuals, you can mainly see changes in this particular chemical shift at the peaks at this particular chemical shifts for the trauma group of pigs. So using chemometrics, you are able to see a clearer visualization of the changes in terms of your metabolic profile, your after trauma being highly separated from the rest of your sample, from your baseline as well as your sham samples. Now, in terms of the OPLS DA S-line plot, this is actually used in order to identify ano ba yung metabolites that are differentiating your trauma samples from the baseline and sham samples. And here we can see, so this stated here, positive y-axis values is downregulated. So that means these compounds are actually low in your trauma injury samples compared to your sham samples as well as your baseline samples. So it is mainly changes in your proteins and acetylglycoproteins, lipoproteins, cholesterol, lipids. So basically, it's mainly lipid changes as well as protein changes that is observed in terms of the changes in the NMR metabolomic profile. So on the other hand, here is an example. Here is the PCA score plots for your LCMS analysis. So again, we made use of a multi-mode LCMS analysis. So we made use of hydrophilic interaction and reverse phase LCMS. Hydrophilic interaction to analyze the polar metabolites, reverse phase LCMS for the less polar metabolites. And we made use of both positive and negative ionization, MS, for the detection. So again, although we have to make use of a 3D plot in order to see the differentiation kasi mas maraming variables yung involved when we're working with LCMS data. So actually, you can generate over 3,000 or 4,000 different variables in some cases. So here you can see also the differentiation of your after trauma samples from their baseline and sham samples. So from here, you can see na meron talagang distinct metabolic profile yung after trauma injury that is present in these pig subjects. So this is just to show the different differentiating metabolites in terms of the LCMS analysis, and you can see the diversity of metabolites that has been tentatively identified. You can see changes in your saturated and unsaturated fatty acids, oxo and hydroxy fatty acids, in your purines and pyrimidines, in your amino acids and derivatives, organic acids, as well as in your lipids. So this Venn diagram is just to show that while there are certain compounds that has been identified in one, two, or more different ionization modes, there is actually a greater diversity in those that are identified in separate techniques. So this is just to highlight the need to make use of complementary analytical platforms in order to increase the coverage of your metabolome. So this is to show the different pathways that has been affected. So mainly it's the glycerophospholipid, purine, pyrimidine pathways, as well as some of the amino acid pathways. This is just a representation of the different, this is just a combination of the different pathways that has been affected. So I was able to see it or to combine everything in a diagram such as this, and you can see the central role that is played by the methionine metabolism pathway in terms of this response to the trauma injury. We also observed an accumulation of uremic metabolites. So when we talk about uremia or uremic syndrome, it is the accumulation of urea and certain metabolites in the blood due to the presence of renal dysfunction or failure. So this is traditionally associated with chronic kidney disease. This results, the accumulation of these compounds or metabolites is indicative of the presence of possible uremia in the trauma injury pig sample. So from here, you can see certain uremic toxins as well as uremic retention solutes that are accumulating. So this can be indicative of acute kidney injury due to the ischemic shock. And because of this, we see that, okay, there is a possibility that early on after the shock, trauma hemorrhagic shock, there can already be renal dysfunction that is occurring or renal dysfunction and other organ dysfunction that is occurring in the pig model. And so we looked further into certain protein markers of like inflammatory response as well as organ failure. So you have cytokines that are related or that are associated with inflammatory response. So these are responsible for the inflammatory response. And you also have like some molecules such as KIM1 or kidney injury molecule 1 and GAL that are more associated with renal injury. You also have those that are more associated with myocardial injury such as CKMB, HFADP, and C-reactive proteins. So from here, you can see those that are colored are those that are higher in terms of the fold changes after trauma injury. So so fold changes is we obtain the fold change comparing the before and after trauma. So elevated levels, so levels that are higher than 1 are indicative of higher levels of these markers in the after trauma compared to the before trauma. And these asterisk asterisk denotes the significance. So 2 asterisk, this denotes a p-value of less than 0.01. These are Benjamini-Hochberg adjusted p-values. So here you can see that early on there is some indications of the effects in terms of inflammatory response, acute myocardial uh, dysfunction, as well as renal dysfunction in the pig model after injury. So for this one, we just did a correlation analysis trying to identify the associations or relations of the metabolites with these different protein markers. And I just wanted to highlight some of these compounds or metabolites that are highlighted that are significantly correlated with certain for example, here KIM1 and NGAL, which are mainly indicating renal injury. You have 1-methyl uric acid, which is mainly associated with your markers for myocardial injury, such as your HFABP, CKMB, and CRP. And you also have your group of sulfur metabolites that are mainly correlated with your inflammatory response markers, which are your cytokines. And with this, we made use of network analysis. So there is already a paradigm shift. Instead of just looking at pathways, now people are looking at networks. So this is in recognition that these pathways are not existing in itself. It is actually part of this whole network of metabolism. And so there is the interdependence of the pathways, and this is very useful for the integration of omics, of the different omics data. So we made use of correlation network analysis to explain the relationships or interactions present in these protein markers as well as with the metabolites in order to gain novel insights into the biological mechanisms, and in order to easier visualize those observed correlations between these two groups of molecules. So in terms of the succinoadenosine metabolites, this is the subnetwork of the correlation network analysis. And in the immediate network or close network of this succinoadenosine metabolite, you can see the KIM1 and the NGAL that is directly and strongly correlated to this succinoadenosine, as well as other purine metabolites and some methylated metabolites. So actually, if you look at the purine biosynthesis metabolic pathway, succinoadenosine is actually a product that is produced if the formation of AMP and subsequently ATP does not proceed. And this actually, the elevation of this metabolite is actually a bad indication. This actually indicates that there is a failure of the conversion of this adenylosuccinate into your ATP. And ATP is a main energy source in terms of the metabolic pathways. And therefore, this elevation of succinoadenosine can be indicative of the dysfunction of this pathway. Unfortunately, actually, the production of GTP is also co-regulated by ATP, and therefore, not only is ATP production affected, but also GTP. And it has been reported previously that GTP depletion of GTP is actually associated with renal tubular injury after ischemia. So this is just to also support the observed possible renal dysfunction in the model. This is just to show the cysteine methionine network. So you can see mainly your cysteine methionine metabolites as well as your methylated metabolites. And this is very much reflected in the actual cysteine methionine pathway. And this reflects the relevance of the correlation network analysis that we have done. We can see the association or the corresponding, kumbaga, match yung nakikita natin in terms of the network analysis as well as the pathway analysis. This is to show the association of your S-adenosylmethionine and cysteine with the inflammatory markers. So S-adenosylmethionine is actually a metabolite that has been implicated in the modulation of the inflammatory response. And as you can see here, there is a direct relationship of this metabolite with these different inflammatory response markers. This, on the other hand, shows you 1-methyl uric acid that is correlated or associated with your myocardial injury markers such as CKMB, HFABP, and CRP, together in linkage with this 1-methyl guanosine. And here we propose some possible mechanisms by which 1-methyl guanosine is converted to 1-methyl uric acid. And this actually implicates 1-methyl uric acid in possible oxidative stress, apoptosis due to GTP depletion kasi mas mataas yung levels of your guanosine degradation products. And nandyan din yung highly activated inflammatory response as can be observed with increased methylation. So in summary, in this particular application, we have used Metabolomics to obtain a comprehensive profile of trauma hemorrhagic shock using NMR and LCMS. We have identified metabolites that are related to impaired energy metabolism, in particular the depletion of ATP, as well as metabolites that can be related to oxidative stress, the acute phase inflammatory response, and the activation of the inflammatory response. We have also identified the accumulation of uremic retention solutes and toxins that is indicative of possible renal injury. Subsequently, we also used correlation network analysis to gain more insights into the metabolic and inflammatory response to trauma hemorrhagic shock. And from here, we were able to identify potential renal and myocardial injury metabolic markers of this porcine model. And subsequently, we also see the important or the association of your cysteine methionine pathway with the inflammatory response and possible development of organ failure. And so, again, here is the different applications of metabolomics. Again, this is non-exhaustive. Of course, we are very much looking forward to hearing more of your own application, of your own research and how we can apply metabolomics. And up next, Dr. Feka Lingasyon will be speaking about her own application of metabolomics in rice research. And we can also look forward to the presentations of Dr. Emmanuel Garcia and Dr. Rodolfo Sumayaw in the subsequent sessions on their applications of metabolomics in food and clinical research. I just wanted to acknowledge the OST, PCHRD, and the Balik Scientist program for this opportunity to be a Balik Scientist. De La Salle University Manila for hosting me as their Balik Scientist, Professor Drexel Comacho, who is instrumental in establishing this Balik Scientist program for me as well. The LSU SIF staff has always been supportive and is in charge of all the logistics of this webinar series. Dr. Feka Lingasyon, Dr. Mel Garcia, and Dr. Rod Sumayaw for agreeing to be part of this webinar series. And I very much, again, look forward to hearing about your own applications, and hopefully we can also collaborate in the future. Prof. Sam Lee, my previous advisor and boss, and who has been very instrumental in my research into my venture into this research field. So thank you very much. He has given me such independence and freedom to explore my own research interests, and for that I'm very grateful. Associate Professor Shabir Machala and the DSO Demerit team for Professor Shabir Machala is actually my co-supervisor, and they have been a very instrumental part of my thesis because I have mainly looked at trauma injury as part of my collaboration with them. I would also like to thank, of course, all of my collaborators, past and present, that has given me opportunities to explore the different applications of metabolomics. The NUS Environmental Research Institute staff, researchers, and management, past and present, who has been very helpful for me in so many ways in my decade in NUS. So NIRI has been a second home for me. I'm very grateful to them as well. Of course, much gratitude goes to my family and friends, who has been a constant source of inspiration and support. So, and lastly, I would just like to share this slide. This is actually a quotation from the Catechism of the Catholic Church. And actually, this is pretty much reflective of my own journey in this research field. And through metabolomics, I was able to see how intricately interwoven everything is, how the regulation, the coordination of these different metabolic networks and the different molecular levels are. And this just puts me in such awe and gratitude to the Lord for giving me this, for leading me into this path and showing the marvels of His creation in my research work. And for that, again, thank you. That's all. If ever you have any concerns, or if you are looking into applications of metabolomics in your own research areas, please do let me know. How can I be of help? So that's all. Thank you.